Good morning, everyone. It's a pleasure to be here today with all of you. And I think we have a really interesting session to open the day. It's going to focus on signaling mechanisms in pancreatitis and in pancreatic cancer. And it's a real pleasure to introduce our first speaker, Dr. Corinne Bousquet, coming from Toulouse in France. She will give an overview focusing on cancer-associated fibroblasts in signaling underlying pancreatic cancer aggressiveness. Thank you very much, Corinne, for being with us, and the floor is yours.
Hello, my name is Corinne Bousquet. I'm from Toulouse Cancer Research Center in France. And I would like to thank the EPC organizer for this invitation. So we talk today about cancer-associated fibroblast signaling underlying pancreatic cancer aggressiveness. I have no financial to disclose. The pancreatic ductal adenocarcinoma is characterized by its fibrotic microenvironment, which represents up to 80% of the tumor mass. As you can see on this patient tumor slide in pink, the stroma, which comprised an abundant deposition of extracellular matrix protein, which represents a physical barrier inducing stiffness and hypoxia and hypoperfusion. Cancer-associated fibroblasts are the most numerous stromal cells present in the stroma. This secretes this abundant extracellular matrix protein, but also soluble factor, such as growth factor, inducing tumor cell proliferation, survival, and also cytokine and chemokines that induce an immunosuppressive microenvironment. So we are now convinced that it's important to target not only tumor cells, but also the stroma to improve patient survival. There have been some attempts to target this fibrotic stroma, especially targeting extracellular matrix protein or CAF. And the idea is here to normalize the stroma and to favor drug entry and the activity of drug. Also, these attempts were efficient in preclinical studies. They failed in clinic. And for example, targeting the extracellular matrix deposit, especially hyaluronic acid using enzymatic strategy, failed. Also, it was successful in phase 1, phase 2, it failed in phase 3. So why? So we have learned that fibroblasts, as I was telling, are pro-tumoral, so they send signal to tumor cells and to other cells of the stroma, which are mainly pro-tumoral. But back in 2014, we have learned that if we try to block CAF, for example, by inhibiting sonic hedgehog pathway, or if we deplete alpha-SMA for smooth muscle-acting positive fibroblasts in pancreatic tumor, instead of blocking tumor growth, we induce more dedifferentiated tumor, more vascularized tumor which are more invasive. So it came to the conclusion that in addition to tumor promoting function, fibroblasts might have also tumor suppressive function. So tumor promoting, so as I was saying, dialoguing with tumor cells, with the tumor immune microenvironment, and also providing metabolic support. We don't know yet which is the identity of this anti-tumoral CAF. Better understanding of this CAF came from single-cell transcriptomic analysis, which described different population of fibroblasts in the normal pancreas, in early lesion of pancreatic tumor, and also in established tumor. The consensus is now to describe mainly 3 different CAF subset, inflammatory, myofibroblastic, and antigen-presenting CAF. Antigen-presenting CAF are the latest described. They are of a mesothelial cell origin, and also they present antigen. They don't express co-stimulatory molecule, so they cannot activate an anti-tumoral immunity, but rather activate Treg and so are immunosuppressive. Concerning myofibroblastic and inflammatory CAF, we have learned that they are interchangeable. In fact, myofibroblastic CAF are activated upon TGF-beta signaling. These fibroblasts are close to tumor cells. They produce many extracellular matrix protein, so providing a physical barrier to drugs and to CD8 T cell infiltration. Instead, inflammatory CAF are activated through interleukin-1 signaling. They secrete many cytokine and chemokines that attract lymphocyte and myeloid cells. And in fact, we have learned that these cells are plastic, interchangeable, because, for example, the TGF-beta impact negatively on the interleukin-1 signal. So recently, we described very new data showing that this CAF subpopulation might cooperate. In fact, we started with a signature published by Neuzy et al. In 2019, describing from RNA signature obtained from CAF from agriculture that there are at least 4 different CAF subpopulations, 2 main, perostine and podoplanine positive CAF, being linked with the prognosis. So now we have refined this signature and applied them to more than 500 patient tumors, so bioinformatically. We have now described that the podoplanine positive tumors, so tumor enriched with podoplanine positive CAF, are immune infiltrated. And as you can see here, you can see that podoplanine RNA signature are correlated, significantly correlated with infiltration of dendritic cells inside the tumor. Instead, periostine positive tumors, so tumor infiltrated with periostine positive CAF, are rather immune poor, but they present some macrophage, and especially the podoplanine, the periostine signature correlates with infiltration of M2 macrophage. When we compared our RNA signature from CAF with single-cell transcriptomic signature, we could observe that our podoplanine positive CAF resemble the inflammatory CAF, whereas the periostine positive CAF are kind of unique. Also, they resemble the CAF described that are LRRC15 positive and so TGF-beta activated. We showed that the presence of periostine positive CAF in tumors is a marker of poor prognosis. And in fact, if we compare podoplanine negative and periostine positive tumors, you can see that podoplanine positive tumor is highly correlated with immune infiltration. But what is very interesting is that depending on whether this podoplanine positive rich tumor are enriched also with periostine positive CAF or not, you will have a different T cell infiltrate. When periostine positive CAF are also present, we can see a very poor infiltration of T cells, but rather high infiltration of macrophage, especially M2 macrophage. Instead, when tumors are rich with podoplanine positive CAF, but poor with periostine positive CAF, you have kind of anti-tumoral immunity, so presence of CD8 T cells, but no macrophage population. So second, we asked how we can target the CAFs that are aggressive. So obviously the periostine positive CAF that are TGF-beta activated. We went back from published work of this year, showing that CAF or poor prognosis are rather myofibroblast CAF that present an activated signaling of the focal adhesion kinase. So these are contractile CAF. We had also published last year that using a cohort of more than of 120 patients, we could correlate the high FAC activity in CAF with a poor prognosis of patients, meaning that FAC signaling might be important to target the CAFs that are of worse prognosis. So using a patient cohort, we indeed quantify FAC activity by using an antibody recognizing its phosphorylated form. And as you can see, FAC activity is much higher in cancer as compared to normal tissue when we quantify this activity specifically in CAF. And if we compare the prognosis of patients presenting a high FAC activity in CAF as compared to a low activity, we can see that these patients have a worse prognosis, which is not the case when we quantified FAC activity in tumor cells. So FAC activity in CAF is highly related with a poor prognosis. So to prove that FAC activity is important for the protumoral function of CAF, we use a syngeneic model co-grafting orthotopically cancer cells with fibroblasts that express either a wild-type form of FAC or a kinase form of FAC. So when we co-graft fibroblasts with wild-type FAC, we obtain lung metastasis, which was not the case when FAC activity was decreased in fibroblasts. This was correlated in the primary tumor with a decrease of M2 infiltration in tumors that present inactive FAC in fibroblasts. And this was true also in patients. So in patients presenting a low FAC activity in fibroblasts, these tumors were not as much infiltrated with M2 macrophages. Regarding CD8 T cells, again in our murine model, where when we had low FAC activity in fibroblasts, we had increased infiltration with CD8 T cells. And all of this was correlated with a difference deposition of extracellular matrix in tumor presenting high or low FAC activity in the fibroblasts, especially in the tumor presenting low FAC activity in the fibroblasts. We had a normalization of this extracellular matrix deposit. This deposition was parallel to the tumor age. And we couldn't observe, as in high FAC activity tumor, invasion of fibroblasts inside the adjacent normal tissue. So in conclusion, we can say that there are two main fibroblasts, cancer fibroblasts in pancreatic tumor. One side, the TGF beta activated CAF, which are positive for periostin, and the interleukin activated CAF, which resembles the inflammatory CAF and which are also podoplanine positive. This tumor presenting which are rich with podoplanine positive CAF are immune infiltrated. But depending on whether they are also rich in CAF positive or not for periostin, they will present an anti-tumoral good immunity or a pro-tumoral bad immunity, meaning that there is a cooperation between this podoplanine and periostin positive CAF to determine which immune cells are infiltrated in this tumor. Then we show that if we block FAC in CAF, and FAC is mainly activated in TGF-beta activated CAF, we can remodel the extracellular matrix, and from an immunosuppressive tumor immune microenvironment, we can switch to an anti-tumoral tumor immune microenvironment, meaning that it could be efficient to combine FAC inhibitor in the future with immune checkpoint blocker. So to finish, I would like to thank the team, my team at CRCT, so in Toulouse, in France. I would like to thank my collaborator, especially the clinician, and also my international collaborator and my sponsor. And I would like to thank you for your attention.
Thank you very much, Corinne, for this amazing talk. It's always really nice to see an update on how things are moving forward with the tumor stroma and pancreatic cancer. I think some questions are coming in the chat, but I think was a really clear overview. So thank you.
Thank you.
One question from Federica Mangilli. Do the two CAFs subtypes or subpopulations derive from the same original type of CAFs? And are there specific molecular signatures defining them?
So concerning the origin, so as I was saying, the antigen presenting CAF probably come from a different origin, as it has been published like these last weeks. So they might come from mesothelial cell origin. Instead, the inflammatory and myofibristic CAF, apparently they can be interchangeable. So probably they come from the same cell origin. Also, it's it's difficult to really affirm this. So I don't have any proof. We know that there are some myofibristic CAF populations that come from pancreatic stellate cells. This was published also this year. And because inflammatory and myofibristic CAF are interchangeable, probably at least some subtype may come from the same origin, yes, cell origin. And concerning the signature, the question, our signature were derived from both CAF primary culture and also we refined this signature with some bulk RNA-seq coming from patient tumor, which we were enriched either with periostin or with podoplanine. So the idea here was to describe and to have signature, molecular signatures that we could then apply to bulk RNA-seq to do some deconvolution analysis and trying to find out in which tumor we have this periostin positive or podoplanine positive CAF population.
Yeah, thank you very much. Yeah, I think you're doing a fantastic job in there. It's really fascinating. So congratulations again, and more questions are coming. I think Professor Real is submitting one question that I think is really important. Do you know whether the CAFs that we are finding in the metastatic lesions are different from those in the primary tumor?
So we didn't check in our study. We didn't check in metastasis yet. So it's difficult to say. We didn't study the subtype in the metastatic place. But what I can say from my experience is that when the metastasis, at least in the mouse models, when the metastases are still small, they are not so enriched in CAF, but when they get bigger, they are really becoming fibrotic and enriched with CAF. But now we didn't study this subtype yet in the metastasis. But it's very nice and interesting question. It would be important to look at this, since depending as we showed that whether CAF express or not podoplanine or periostin, patient could receive or not immunotherapies. And because for metastatic patients, for example, we have only this liver biopsies, it might be interesting to check whether these subpopulation are present, and if we could stratify patients based on this CAF subpopulation to give or not immunotherapies. That might be very interesting.
I think it's a really, great question and really key to better understand also relapse and prognosis of patients. I was attending a forum a few days ago here in Oxford, and was a big question regarding studying metastatic disease, but the different researchers were mentioning the limitation to access to metastatic tissue, right? Also, it's kind of challenging to get these biopsies, but hopefully in the next years we will see something else ongoing in this field. I think we have time for one quick question. So from Professor Magirle, we'd like to know a bit more on the FAC targeting that you were mentioning. So there is some work published by Kevs, Hodivala and Dielke, saying that FAC also plays a role in neuroangiogenesis in cancer. Can you comment a little bit on that, please?
Yeah, FAC is not like FAC activity is not specific to CAF. So FAC is activated also in tumor cells and in endothelial cells. And we think that it's a good target because in each cell type where it's activated, it has like pro-tumoral action. So the inhibition, like when we treat a patient with FAC inhibitor, of course, we will act in all these cell types. So it's important to verify that the activities, when you block it, you will have a benefit, right? So because this activity is really pro-tumoral in each cell type where it has been studied, we think it's very important to target it. But yes, we have to take into account the activity of FAC in all cell type, tumor cells, and all in the whole microenvironment. Yes, because otherwise we can have side effects, right? So we will see the clinical trial ongoing for in patients. So we will see if we can have a nice result with them.
Thank you very much, Corinne. I think we are all looking forward to see the clinical trials. And yeah, thank you so much for this.
Thank you for this invitation, and thank you for the Congress.
Yeah, and thank you for this fantastic discussion. Sadly, I think we have a lot of different questions, but we need to move forward to our second speaker.
Okay, I can get the question by mail and try to answer.
Thank you so much. Thank you so much. And now I give the word to Professor Paco Real.
Good morning, everybody. So it's my pleasure to be here with you and to introduce Professor Julia Mayerle, who is going to talk about a very interesting topic, complex lipids in pancreatic cancer, a neglected field. Julia comes from the LMU University in Munich. So please, Julia, go ahead. Thank you.
Ladies and gentlemen, it's a pleasure to present to you today on complex lipids in pancreatic cancer, a neglected field. It's a pity that we are all not able to come to Kiev, but hopefully this will happen in the future. Thanks to Natalia Gubergritz for organizing. So why sphingolipids? Lipids, mostly for technical reasons, have been in the shadow of other omics technologies. Lipids are small molecules of enormous chemical diversity and not restricted to chemically distinct building blocks. They build membranes. They play an essential part in our mammalian organism. What we learned from lipidomic studies is that mammalian cells use 5% of their genes to synthesize lipids. Although structurally, lipids are composed of few building blocks, they have the potential to generate 9,000 to 100,000 different molecular species, all being able to signal. So when it comes to sphingolipid-mediated mechanism promoting cancer, and you look at this wheel, that you find all relevant processes in carcinogenesis being targeted or affected by sphingolipid-mediated mechanisms. So there's evading growth suppressors, avoid immune destruction, the replicative immortality, pro-tumorigenic inflammation, invasion and metastasis, apoptosis, angiogenesis, and also energetics and proliferative signaling. So the sphingolipid metabolism is highly compartmentalized. So we have the de novo pathway in the endoplasmatic reticulum. We have the sphingomyelin pathway, mainly functioning at the cell membrane. And we have the salvage pathway, which can be found in the late endosomes and lysosomes. All of them work together to build ceramide and also sphingosine 1 phosphate as the most important signaling molecules or ceramide moieties to affect cellular responses such as cell growth, viability, differentiation, and senescence. Important enzymes here, just to give you an example, would be acid sphingomyelinase. So the ceramide sphingosine and sphingosine 1-phosphate rheostat is a double-edged sword. So sphingolipids form either ceramide or sphingosine 1-phosphate, and sphingosine 1-phosphate functions as a pro-tumorigenic signaling molecule, while ceramide molecules in many instances function as anti-tumorigenic molecules. How can these rheostats work, and how is this intricate balance maintained? So from sphingomyelin, SMPD1 or acid sphingomyelinase produces ceramide. Ceramide can also be produced in a de novo synthesis. And from ceramide, sphingosine 1-phosphate is generated by an enzyme called SH1 or acid ceramidase. Most likely, SH1, and I will show you data on that, is a tumor promoter. And what we found in our recent data on PDAC is that SMPD1 functions as a tumor suppressor. The sensitivity of cancer cells to chemotherapeutic agent is a function of the rheostat, with higher ceramide levels supporting treatment response. So when you perform an integrated metabolome landscape in PDAC and chronic pancreatitis in mice, and you study serum and tissue, you find that in serum there are a number of important signaling pathways being upregulated, while few are downregulated. If you look at tissue, you seem to observe the opposite effect. A number of pathways are downregulated in tissue, while and mainly in the opposite way as found in serum, and few are upregulated. If you integrate these landscape, you can see that there's clustering. And you can see that tissue and serum, especially, for example, here in cluster, in the green cluster, so that is cluster 4, you see that they entirely behave in the opposite way. So you can find a lot of anti-tumorigenic ceramides like C16, C17, 18 with different chain lengths here. Especially in the tissue, upregulated in the tissue, while it's downregulated in serum. You also can see pro-tumorigenic ceramides, especially in the pink cluster, where they are downregulated in serum and downregulated in tissue. So what you see is that in tissue, there is a deranged balance, and you find more sphingomyelin and less ceramide functioning in a response to a pro-tumorigenic behavior. So tissue has significantly decreased ceramide metabolites, whereas sphingomyelin did not show significant change compared to serum. So what are ceramides? Ceramide is not a single moiety, but it consists of multiple different moieties, and they can be distinguished due to their chain lengths. So they also have different functions, like C18, to give you an example, is anti-proliferative, or C20 fosters apoptosis, or C24 is involved in chemosensitivity. So all of those function as anti-tumorigenic. Others, like C14 being involved in proliferation, or 16 being involved in invasion and metastasis, function as pro-proliferative. So how are they built? They can either build by ceramide synthetases, and we know 6 enzymes, and those enzymes are specific for a specific chain length in ceramides, or they can be they are built by SMPD1, so acid sphingomyelinase, which is bound to the plasma membrane and can also be found in the acidic compartments, and which actually accounts for at least 50%, but up to 90% of the building or generation of cellular ceramides. So what is the role of SMPD1 in pancreatic cancer? Well, if you just look into this TCGA data set, then you can see that high expression of SMPD1 is associated with a significantly increased survival in pancreatic cancer. If you study resection specimen, again, and if you look at the overall paraffin section, then high expression of SMPD1 is involved with a prolonged survival. If you look at the aspect data, and that allows you to do multiplexing in roughly 400 patients and then validated in a smaller group from our own hospital, then you can see that different cells, different cell types do show different expression of SMPD1. And I know that's pretty small, but you can see from the dots here that different cell lines, like this is SMPD1, is expressed differently, for example, in macrophages or cancer cells or T cells. And if you look for SMPD1 expression in tumor cells, then you find a decreased expression or decreased survival in those with SMPD1 low expression, resembling the TCGA data. However, if you just look at the expression of cytotoxic T cells, just to give you an example, then you can observe that the survival of low SMPD1 expression is higher. So tumor cell expression of SMPD1 is associated with an increased survival in PDAC, while loss of SMPD1 in immune cells results in prolonged survival. And just to give you an example why that might be important, inhibition of SMPD1 in platelets prevents metastasis formation in melanoma. So the cellular context is really important to understand sphingolipid metabolism in cancer. So when you look into cell culture and you study pancreatic cancer cells, and you're looking here at kappa and PA2S and PA2T, then you see that SMPD1 knockout cells showed an increased proliferation, invasion, and autophagy, proving that SMPD1 acts as a tumor suppressor in pancreatic cancer cells. If you look at animals, crossbreed with KC animals, then we observed that 50% of the animals developed metastasis at a humane endpoint versus none in the KC wild-type animals. So it seems to be that SMPD1 is involved in metastasis formation. When you look at human cell lines, what you can observe there is that gemcitabine sensitivity is affected by SMPD1. So the knockout results in an increased sensitivity, while it does not have an effect on 5FU. That is also true if you study the SMPD1 cathepsin D ceramide axis. So again, in the ASPECT study, in 362 patients, we studied cathepsin D as a lysosomal enzyme and aspartate protease in the lysosomal compartment, and you can see that upregulation of cathepsin D is leads to decreased survival. So adjuvant gemcitabine and what we also found detected was that adjuvant gemcitabine was less effective in patients with high cathepsin D expression. So what we also found is that acid sphingomyelinase, so SMPD1, deletion leads to upregulation of cathepsin D. And to give you a schematic explanation for that, if you have high levels of cathepsin D, acid-sphingomyelinase is blocked, and therefore the sphingomyelinase in the lysosomal membrane is no longer processed to ceramide, and that leads to leaky membranes, and that gives you a proliferation signal survival and also affects migration and invasion. So you can restore that. So if you look at the acid-sphingomyelinase activity, then you delete or inhibit acid-sphingomyelase inhibiting, then you can actually reverse the phenotype, and the cells are again effective or can be killed by gemcitabine, even in gemcitabine resistant cell line. So there seems to be a ceramide acid-sphingomyelinase cathepsin D axis that is regulated through an intercellular ceramide level by an autocrine loop. If you do metabolic stratification of pancreatic cancer, then you can detect 3 different subtypes of pancreatic cancer, characterized by differential abundance of lipid metabolites. You can also find 3 different metabolic programs which are differentially expressed in these different subtypes, and they are co-metabolic programs stratifying PDAC metabolic subtypes, and they are depicted by the sphingolipid dependence. These subtypes are also correlated to survival. So we not only know subtypes such as classical or basal subtypes, which are defined by a genetic structure, but we also know subtypes which are characterized by sphingolipid metabolism. So if you look for the expression of ASH1 and SMPD1, and I remind you, those are the enzymes involved in the S1P rheostat, then the sphingolipid metabolism associated genes show differential expression in PDAC. SH1 is upregulated and SMPD1 is downregulated in the basal subtype. And the basal subtype shows significant downregulation of this metabolism, and but high expression is associated with prolonged survival, with basal subtype confirming SMPD1 again as a tumor suppressor in human pancreatic cancer. You can also use a plasma metabolic signature for differential diagnosis. You know that symptoms of PDAC and chronic pancreatitis are pretty unspecific and overlaying. So currently we don't have a good marker to detect pancreatic cancer in chronic pancreatitis patient or exclude pancreatic cancer. So in an intricate study with roughly 900 patients in 3 different steps, we developed by elastic net regression and then fetal reduction AI metabolic signature consisting of 12 metabolites plus CA99. Out of those 12 metabolites, the biggest numbers consist out of sphingomyelins and complex lipids. You can see that the I-metabolic signature can clearly discriminate between pancreatic cancer, chronic pancreatitis, and non-pancreatitis controls. And our metabolic signature would have correctly diagnosed 15% more patients with chronic pancreatitis and would have identified an additional 12% with PDAC, which would have been missed if you would have used CA99 alone. However, measuring 12 metabolites plus CA99 is a huge effort, and therefore we tried in a pre-analytic and analytical confounded performance analysis to reduce the number of metabolites for prediction to 5. And what you can see here is that the I-metabolic signature and those 4 metabolites plus CA99 perform equally well after being trained by a base ML model and an iterative ML model using machine learning. So this signature for sure would be cost effective and showed matching performance to a previously identified signature. Moreover, looking for confounders and to understand whether there would be transferability in lab routine, we looked for the different collection tubes, found no influence. We looked for short-term storage. We looked for plasma storage at -20 degrees. We looked for the influence of shipping, hemolytic plasma, lipemic plasma, and common co-medication, and could not find any influence. And the only influence we could determine was the influence of non-fasting that gave us an increase in false negatives, and icteric plasma, which also gave us an increase in false positives. So in summary, mammalian cells use 5% of their genes to synthesize lipids. Although structurally, lipids are composed of few building blocks, they have the potential to generate 9,000 to 100,000 different molecular moieties involved in essential processes in health and disease. Targeting the sphingolipid metabolism as a therapeutic strategy in cancer treatment is promising and novel. In PDAC, metabolic subtypes and metabolic programs associated with cancer prognosis have been identified. SMPD1 acts as a tumor suppressor, is responsible for gemcitabine resistance, but its role is dependent on the cellular context and somatic mutations. SHA1 as a targetable counterplayer of SMPD1 could be therapeutically exploited, and a sphingolipid signature as diagnostic non-invasive tool to exclude PDAC in high-risk cohorts is established and should be able to be used in the near future. There are many more enzymes and pathways to be studied in lipid biology, and I guess that will make a huge difference to pancreatic cancer treatment in the future. And with this, I'd like to thank all my co-workers, especially Ujjwal Mahayan and Ahmed Al-Nadsha, Didem Saka, Clea Rico, and Shi Li for supporting me on this project and doing all the experiments in the lab. Thanks for your attention.
Thank you very much, Julia, for this very interesting discussion and for presenting this new data. We have time for just one question. There are more questions to Julia that she will answer separately. Gabriele Capurso wishes to know how much plasticity to the metabolic lipidomic subtypes have and whether the more aggressive phenotype can somehow be reverted, at least in vitro.
Well, then, Gabriele, we believe that it should be possible, and we are currently working on that. You see that the gemcitabine resistant cell lines, if you block acid sphingomyelinase activity, then the phenotype can be reversed. So yes, there is plasticity, whether especially the subtypes, like in the classical or basal, so the metabolic subtypes can be entirely reversed. We don't know yet, but we are. That is a project which is ongoing in our lab.
Thank you very much, Julia. Unfortunately, we have to move on to the next talk. Thank you again, everybody, for the questions.
Thank you very much, Professor Real and Professor Majorle, for the discussion. And now we move to the last talk of the session. It's a pleasure to introduce Professor Hannah Algi, who coming from Munich, who will be giving an overview on pro-inflammatory signaling pathways in pancreatic cancer. The floor is yours. Thank you.
So, ladies and gentlemen, it's my great pleasure to be given the opportunity to be part of the virtual EPC meeting in Kiev. We all would have preferred to visit our colleagues in Ukraine, but due to the situation that makes us all very sad, we are only able to meet you virtually, and our hearts are with our colleagues in Ukraine. So these are my financial disclosures. And I was asked to give an overview on the transition phase of pancreatitis and pancreatic cancer. This is a very hot topic and highly discussed both in clinical but also in scientific communities. Understanding the interaction of inflammation with cancer in the pancreas will potentially allow us to identify novel therapeutics targets in the disease. that are either treated only symptomatically, like chronic pancreatitis, or do have limited chemotherapeutic options like pancreatic cancer. Now, as a matter of fact, numerous association studies of CP and PDAC were published by giants in the field of pancreatology over the last decades, all of which revealing a strong relation between both diseases, with being the strongest in those patients with hereditary chronic pancreatitis. And this is shown on the left-hand side with the meta-analysis that I received from the paper in 2017. The association of inflammation and cancer is not new in the GI tract, and we know that from many other diseases. But how are the molecular underpinnings of the transition from chronic pancreatitis to pancreatic cancer? So a landmark study in basic science was provided by Mariano Barbacit's group, who demonstrated then that in the context of oncogenic activation of KRAS in mouse model, inflammation in the gland induced by repetitive injections of cerulean accelerated the malignant transformation. However, detailed mechanisms were not clear and are still not really clear. If you allow, I would only focus on my group and our data to demonstrate our contribution to the field without willingly neglecting or despising the work of others that were also of paramount importance to the field. My group has a strong focus in understanding the molecular mechanisms of pancreatitis in various models of acute pancreatitis using mouse models or human scale models of pigs in pancreatitis. Our focus is not only limited to local inflammation, but also to systemic implications of the disease. Mouse models of chronic pancreatitis shown on the right-hand side of the slide were also established in our group following repetitive injections of cerulean. Particularly in chronic pancreatitis, models are lacking allowing the analysis of the transition phase of inflammation to malignant transformation. Following the accumulating evidence that next to intracellular or intraacinar activation of digestive enzymes, integrity and functionality of organelles, particularly of mitochondria, are key in, for example, ethanol-induced chronic pancreatitis and potentially for the transition phase. We reasoned whether interfering with autophagy, a central regulator of organelle recycling, will provide us more insights. And Nina Diakopoulos embarked on this question and has deleted ATG5, an important regulator of the multimeric process of autophagy, specifically and in a time-dependent manner in the pancreas. And in fact, deletion of ATG5 significantly impaired the process in the pancreas, but not in other organs, as shown on the right-hand side with the Western blot, and also with the electron microscopy picture showing that the autophagosome formation was impaired. Phenotypically, Nina has observed that this inactivation led to a time-dependent destruction of the pancreas, exocrine pancreas, with mitochondria swelling and dilated endoplasmatic reticulum. In the pancreas, clinical or critical features of chronic pancreatitis, such as fibrosis, apoptosis, and necrosis, along with the accumulation of macrophages, could be detected. So signs of a model of chronic pancreatitis, spontaneous chronic pancreatitis based on genetic deletion of ATG5. And yet, Nina and Kivanch did not observe progression to cancer unless in the context of oncogenic activation of KRAS. In this setting, it seemed that tumors that evolved out of this ATG5 deficient pancreas were more aggressive with significantly higher metastatic load. And this was partially due to the concomitant inflammation in the organ. Interestingly, accumulation of macrophages seemed to occur even in spontaneous KRAS-induced carcinogenesis in the pancreas. Marina Lesina, a very hardworking and successful scientist from Odessa in Ukraine, has shown a couple of years ago that macrophages in the pancreas accumulate as soon as PANIN1 lesions appear. And this accumulation increases over time and during progression to cancer. And these macrophages also seem to produce pro-inflammatory cytokines such as IL-6, which is involved in the activation of several pro-inflammatory pathways, predominantly of the GP130 dependent STAT3 signaling cascade shown on the right-hand side of the slide. Interestingly, this phenomenon of IL-6 in macrophages can also be detected in acute pancreatitis, an observation Sang Hong from China described in my group in the Journal of JCI in 2013. So Marina reasoned on the contribution of this phenomenon in pancreatic carcinogenesis, with various models either deleting IL-6 or complexing of IL-6 with the GP130 or the deletion of the downstream effector of STAT3. She could clearly and convincingly demonstrate that this pathway is instrumental in carcinogenesis, as every single genetic alteration significantly inhibited the progression of pannin lesions to cancer. To the converse, the genetic inactivation of the endogenous inhibitor of the pathway SOX3 accelerated carcinogenesis with the occurrence of early cancers and metastasis in a time that we do not observe this in the mouse model. To demonstrate the relevance of this concept, Marina, together with our pathologists at the Technical University of Munich, analyzed human specimens and revealed that the critical components of this concept and pathway are replicated in the human disease. Thus, the good thing about this concept is that it is therapeutically relevant, with compounds being available to interfere with this signaling pathway. And in fact, this concept has led to numerous clinical trials that I cannot discuss with you due to the given time frame. Sonia Werman, who is now at MD Anderson, took over this concept in my group and elaborated in more details the relevance of interfering with this pathway. Interestingly, the extent shown on the right-hand side of the slide, the extent of the pro-inflammatory pathway STAT3 activation is strongly correlated with the survival of patients with metastatic pancreatitis. Thus, in a first approach, Sonia treated PADAC mouse models with the JAK1 and 2 inhibitors that are readily available in the clinic using genetically engineered mouse models. And in fact, she demonstrated that the combined inhibition of this pro-inflammatory pathway through ruxolitinib in combination with gemcitabine decelerated the progression of the tumor growth shown on the right-hand side with the slide. Interestingly, this pathway seems to be also crucial for pancreatitis, and its inhibition ameliorated the extent of inflammation in the pancreas, again showing that inflammation in the pancreas and cancer do share common pathways that are relevant for therapeutic interventions. So the impact of this pathway on the morphology of pancreatitis of pancreatic cancer is far more complex and can be exploited on several levels. Immune therapeutic approaches such as PD-L1, PD-1 inhibitors in PADAC are not very successful in the clinic, even in the setting of MSI-H or MSI-high tumors. One of the biggest obstacles seems to be the relatively low abundance of immune cells in the tumor due to the strong desmoplasia. Marina has observed that IL-6 treatment increases the influx of immune cells and of T cells, and that the combination with PD-L1 induces the regression of the tumor when combined with gemcitabine that usually has no effect on tumor growth, providing a concept how the efficacy of immune therapeutic agents like PD-L1 inhibitors can be increased in pancreatic cancer. We are currently working out this concept in more detail to eventually transfer it to a clinical trial. Our interest in this pathway brought us to another interesting observation that Dietrich Ruß, a surgeon in Freiburg, elaborated in more details. The phosphatase SHP2 is critically involved in regulation of the activation of this pro-inflammatory pathway STAT3. However, when we delete SHP2 in the pancreas specifically, that did not lead to enhancement of STAT3 activation, but abrogated the activation, an expectation that we did not anticipate. More strikingly, that leads to a strong and robust inhibition of tumor development, even in the absence of p53, the most important gatekeeper in tumor biology. This strong effect was associated with an unexpected downregulation of the KRAS signaling signatures. Very likely, SHP2 is therefore critically required for growth factor receptor mediated signaling, even in the presence of KRAS mutation. The therapeutic relevance could be confirmed on 3 levels: in genetically engineered mouse models demonstrating that SHP2 inhibition is sufficient to robustly induce regression of tumors, which was not only true for the genetically engineered mouse model shown here with the MRI scans, but also for organoids, patient-derived organoids, and patient-derived xenografts. In a more preclinical setting, Katrin, Alex, and Marina elaborated the combination of ERK and SHP2 inhibitors, showing that the continuous treatment strongly induced the regression of the tumor macroscopically, and in PET CTs, we identified surrogate parameters for the response of tumors to this combination therapy. So all these concepts are currently evaluated in a phase 1b trial, the so-called SHERPA trial, together with René Bernard's group in MKI Amsterdam. The SHERPA trial has a part A and part B, involving in part A all comers with KRAS mutation and pretreatment, and in phase B we will expand on pancreatic cancer patients with mutated KRAS. We have already started with the clinical trial and we have enrolled patients so far. So this is one example how we can identify and exploit pathways that are inflammatory and also interacting with oncogenic pathways. But it is more complicated. I started with Mariano Barbacit's study that offered anti-inflammatory compounds as a therapeutic add-on because they can restore oncogenic induced senescence. But it is more complicated because Marina has focused on the role of central mediator of inflammation in cancer, which is the NF-κB pathway. And surprisingly, she revealed that the critical subunit of NF-κB, REL-A, is instrumental in inducing oncogenic mediated senescence via CXCL1 and CXCR2 loop. The inhibition of REL-A accelerated the tumor progression. So in the middle of the slide, you can appreciate the beta-gal staining that obviously is dependent on the activation of REL-A. So let me conclude. Pancreatitis and pancreatic cancer do share common mechanistic processes. The chronic inflammation may cause oncogenic mutations that are required in the transition phase, and the immune cells play an important role in the transition phase, but also important in tumor maintenance. The transition phase is still very unclear and difficult for therapeutic as well as preventive exploitation. The concomitant inflammation in the pancreas contributes to tumor maintenance and can be considered as a therapeutic target. We get more insight into the transition of inflammation to cancer, but together with some others, we made some progresses, but it's still very complex, and the complexity is nicely shown by George Miller's group in the sketch on the right side. So due to the time frame, I could not elaborate on macrobiota and translocation of bacteria, which is part of the sketch. So this graph clearly demonstrates the progress we made, but also the topics that are still not clearly understood. With this, I would like to close the talk and would like to thank you for your attention.
Thank you very much, Professor Gül. A very interesting talk. We have time for like 2 minutes for questions. In the meantime, that they are coming in the chat, I think, because you mentioned just briefly in the end about the role of microbiome in inflammation. Can you comment a little bit more on that? How do you see this can guide maybe decisions in the clinic and how we should take this microbiome into account? Thank you.
Well, first of all, we have evidence from different groups that the microbiome impacts tumor development, but also therapeutic interventions. So we are now in the process now, having understood that it plays a role, now we are in the process to understand how we can more decipher its role in carcinogenesis and how we can exploit it therapeutically. So I think there is evidence to go further in details to understand this, but we are not at the point to say, okay, we can exploit it therapeutically right now, or you exploiting it for diagnostic purposes.
Yeah, maybe with a new clinical trial that is ongoing can give some opportunities for sample collection and further understanding, yes, right, of this really nice and interesting study. And I think is right now we are on time, so sorry, but I don't think we don't have more questions in the chat. And I think I would like to thank again all the speakers and the co-chair of this session, Professor Real, for what I think has been a really interesting session. I am sure if you have further questions, you can contact directly all the speakers and continue discussion by email. It's a pity that we cannot go to a coffee break and discuss in person, but I hope we can see each other soon in the next conference. So thank you very much, everyone, for joining, and thank you to all the speakers of the session. Thank you.
Thank you. Bye-bye.
Thank you.